Before we get into the answer, though, briefly, I won't be able to do office hours today because it's for a good reason. Well, it's always for a good reason, right? I am the chapter advisor for our local Society for the Advancement of Chicanos and Native Americans in Science Club, SACNAS, and they're having their first semester meeting immediately after class at noon in Peters Building Room 101. If you're interested, you don't have to be a Chicano or Native American, clearly, to be involved. But if you're interested in participation, which basically gives you lots of support in identifying resources and preparation for careers and so forth, you're certainly welcome to come with me right after class across to Peters Building 101. So that's what I'll be doing instead of office hours today. Normal office hours will resume on Wednesday, and as usual, if you need to set up an individual meeting with me, I'm happy to do that. Okay. So how did you know this was an indel? Because
he has the exact same sequence, only with the addition of a C.
So this is the really important part. It's the same sequence. Right? So I'm circling parts of the sequence that are identical in the two chromosomes. There's just one change. It's the addition, insertion of a nucleotide. If you were looking at this and thinking, oh, here we're looking at a G and a C, and that's a difference, and that's a single nucleotide polymorphism, the reason that's not the way to do it is because of what's circled here in red. That we have the same sequence of the two chromosomes, and that's how we know that this is an insertion of a new nucleotide, not a point mutation or a single nucleotide polymorphism. Another way to say that is, if there's a change in the size of the chromosome, as you can see up here, one's longer than the other, that's the clue for indel, insertion or deletion, not SNP, not a single nucleotide polymorphism. Yeah, please. So we're going to talk about microsatellites today, and I will definitely do that, absolutely. So any questions about single nucleotide polymorphisms, indels? So microsatellites. Indel or single nucleotide polymorphism? It's a type of, microsatellite is a type of indel. So microsatellite is a fancy word for a short sequence of DNA that's repeated over and over and over again. For example, let's take this case. Let's say this G and the A were repeated multiple times. If you had a microsatellite in your DNA sequence, you would have, as in this example, the same sequence on both sides. You'd have a TAT on one side, and you'd have a CAT on the other side of the microsatellite. So what am I going to draw in here? One chromosome just has one GA. That's one repeat of that dinucleotide. Somebody might have 3 copies of that, GA, GA, GA. So we've got 1 copy of GA on one chromosome, we've got 3 copies on the other. And normally, there are lots and lots of places in our genomes we'll talk about today where there are tens or hundreds of repeats of 2 nucleotides in a row. GA, 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 and that makes up a lot of our genomes, our 300 billion base pairs of DNA. A lot of that is basically what happens, we're pretty sure what happens, scientists, that when the chromosomes are copied, when DNA polymerase, the enzyme that actually synthesizes the DNA, gets to one of these regions where it's the same nucleotides over and over again, that the polymerase, this is anthropomorphizing, right? This is me putting personality on an enzyme which has no personality or brain. It stutters. It gets lost. It's like, how many copies of this dinucleotide did I already make? It sort of loses track of how many copies it's produced, and it can add or delete units from this microsatellite. So some of us have 27 copies. Other of us, if you look at your chromosomes, you might have 31 copies of a GA, a TC, something like that.
Does that affect the protein that comes out of that particular gene?
So if this microsatellite occurs in a gene, then it would affect the protein that it encodes. Lots of these microsatellites are found in between genes. So it doesn't matter whether I've got 19 copies or 31 copies of an AT microsatellite. It doesn't really affect me. But there are lots of cases where it will. Last thing, what's the nomenclature for this? 3 repeats of a GA. Right, with parentheses usually. So we'd parenthesize what the repeat unit is, a single unit, and then put a 3, usually as a subscript, but not necessarily after it. So this would be an individual that has a GA3, and if this was 2 chromosomes from the same individual, their genotype would be GA1/GA3. They have 1 repeat of GA on 1 homolog, 1 copy of this chromosome. And they have a repeat of 3 on the other homolog. I realize I never give you my answer to this exercise. And what I'm going to do is I'm going to post it online. I have a video of me working through how to solve it, so you can use that as a reference if you want. My answer is not the correct answer, as we discussed last class. There are multiple ways to do this, and hopefully some of you did actually try rearranging the order of events you used, deletions, inversions, and so forth. So you'll have the answer to that digitally sent to you. One last point before we move on to today's real topic, which is mutation. A couple of things that I didn't get to cover 2 classes ago. So this is review. I wanted to mention that the blown up view of the nucleosome, which is defined here, 8 histone proteins, which we discussed. This is what they look like when they all come together in a blob. So those 8 histone proteins make the nucleosome when DNA wraps around it about twice, as is shown up here. The thing that we didn't get to in class was different types of chromatin. So does anybody remember the definition of chromatin? DNA plus nucleosome, so DNA plus protein. So in both of these pictures, the bottom left and the bottom right, this one here especially, we're looking at chromatin. That's DNA wound up in all of these nucleosomes, the histone proteins. And these 2 pictures are meant to make an important point. There are 2 types or 2 classes of chromatin, euchromatin and heterochromatin. This is just definitions, so I'm going to go through it pretty quickly. Except I'm doing it backwards. Heterochromatin on the left, euchromatin on the right. The difference here is the difference that you see. On the left, the DNA is really packed densely with proteins, and as we discussed, on the right, all the proteins have been taken away from the DNA down here. That's an extreme example of euchromatin or open chromatin. So if you were a protein and you were trying to access a gene and read it, like you were RNA polymerase, and you were going to go bind the DNA sequence, read it, and spit out a transcript, an RNA molecule, which type of chromatin would be easier for that protein to do its job? Heterochromatin or euchromatin? Why? The DNA is more accessible. It's not tightly wound up in proteins. So on the euchromatin side, this is what DNA looks like on the right when the cell is in interphase, right? Not when it's going through cell division. So this is what the chromosomes look more like. Euchromatin, when the cell's actually doing the business of the cell. Genes are being transcribed and translated, turned into proteins. The cell is growing. The cell is doing its normal jobs. When the chromosomes are all packed up in heterochromatin, it's difficult, and in fact, usually during metaphase, there is no transcription because that DNA is wound so tightly together in proteins that nothing can actually access that DNA. No proteins, no enzymes. So two important terms to know, euchromatin and heterochromatin, and how they relate to how the chromosome is compacted and what can or can't happen during that process. This is one of my favorite pictures in all of genetics. This is again from a muntjac, as it says down here at the bottom, that you can't see yet. Oh, good, now you can. These are chromosomes that have been stained according to which chromosome they are. Remember, there are 3 different pairs of chromosomes in muntjacs. They're 2n equals n is the number of different chromosomes there are. So how many different colors of chromosomes are up here? 3. So n is 3. So twice n is 6. Muntjacs are diploid. They have 2 copies of every chromosome. You can see up here, these ones down here, where you actually observed the chromosomes as discrete units, when was that picture taken? During the cell cycle. That's, you said it, say it louder.
Metaphase.
That's metaphase. Remember, a couple of classes ago we talked about if you see a chromosome and it looks like we think of a chromosome, those are taken during metaphase, as it says right there on the slide. So they've done the exact same, this is why I love this picture. They've done the exact same thing. They've stained the DNA of the different chromosomes in that picture on the top. That's an interphase nucleus. That's what your chromosomes look like when, and muntjac chromosomes as well, what they look like when the cell is not preparing to divide. So which one's euchromatin and which one's heterochromatin? Euchromatin's on top, interphase. The chromosomes are not compacted into metaphase compact chromosomes. They're open and accessible and able to be transcribed, the proteins then translated and so forth. What's interesting about that picture? Do you notice anything that maybe is unexpected in that interphase nucleus?
They're dispersed all around, not clumps, not.
So the chromatin, or the chromosomes, the DNA fills the nucleus. But what's another way that could look? What's another way you could draw in how chromosomes might expand and fill the nucleus during interphase?
Spaghetti almost?
Yeah, so they could be like, they could all be tangled together. What's not happening here?
They're sort of separated.
They've expanded, but they've expanded in their own regions of the nucleus. And when chromosomes are in interphase, they don't tangle all around each other. They still stay in local regions. Each chromosome stays in a local region of the nucleus. And that's really important, it turns out. In the last 10 years or so, geneticists have discovered that that has a lot to do with which genes get used and which ones don't. Are you at the edge of the nucleus, or are you in the middle of the nucleus? Chromosomes that express sets of genes tend to be clustered together in the same parts of the nucleus, so this is not random. It's actually meticulously placed cotton candy chromosomes. The reason that knowing about euchromatin and heterochromatin is important is because cytogeneticists, the geneticists that study chromosomes, use this technique, several of these techniques, chromosome banding, to identify the different chromosomes. And you've seen loads of pictures of these. I don't know if you've really appreciated what happens, but there's particular patterns of dark and light bands. You don't normally see this when you look at chromosomes. If you just look at chromosomes, you see nothing. We have to actually stain the DNA to be able to see it with our eyes. And G bands are produced, this is just an example, there are many types of chromosome banding. They're called G bands because they're stained with a dye called Giemsa stain dye. And that dye specifically binds to AT-rich regions of the chromosome. That is, regions of the chromosome that have lots of A's and T's. So it's interesting. Our chromosomes from left side to right side, from top to bottom, however you're looking at them, aren't ATGC, ATGC. They're not equally 25% each of the 4 nucleotides across the entire chromosome. there are regions of our chromosomes that have more G's and C's, and then there's some regions that have more A's and T's than others. And that's why we can see these sorts of banding patterns when we stain chromosomes with certain chemicals. And it just happens that it's useful for us for identifying the chromosomes because each chromosome, this is the key point, each chromosome has a unique banding pattern. And that's how cytogeneticists can stain these chromosomes and then pair them like this to look at the two homologs together. Yeah.
In the book, it said that the light bending pattern meant that the genes were active, and active genes, and the dark stained ones meant inactive, so that means that the AT are inactive always?
So here's I'm glad you asked that question. How does the color of the band relate to activity, euchromatin versus heterochromatin? Critical point. AT-rich regions are usually heterochromatic. That is, genes, it tends, and we'll learn about this more, GC-rich regions of the genome tend to have genes in them. Regions that are mostly A's and T's tend not to have genes in them. They're gene-poor regions, so they tend to be heterochromatic. They're all condensed most of the cell cycle because there's no genes there anyway, so why would the cell bother decondensing into euchromatin? There are no genes there to be read in the first place. So G banding, those dark bands are the AT-rich regions, gene-poor regions. There's a different type of banding called R banding, and I don't want you to remember all these details, I just want you to have heard it once. R banding, the R stands for reverse. That stains, what do you think? Yeah, the C's and G's. Which means that if you're looking at an R-banded chromosome, the dark regions are the gene-rich or euchromatic regions. So you just have to know which stain you're looking at. Because G banding and R banding give you the opposite banding patterns. One shows you euchromatin, one shows you heterochromatin. You can't just look at a banded chromosome without any information and know what that dark banding pattern means. You have to know if it's G banding or R banding. Or, for example, down here on the bottom right, a different type of banding, C banding. C for centromere. It's a stain that only reacts to centromeres. And that's how we can identify the dark spots are all centromeres. That's an introduction to 3 different types of chromosome banding. There are lots. The key use is to be able to identify the banding fingerprint, so to speak, of a chromosome. Each chromosome will have a different pattern of bands.
So G banding applies to the AT-rich, which also happens to be heterochromatic, is that?
Correct. So the definition is AT-rich regions are usually heterochromatic. GC-rich regions of chromosomes tend to have a lot of genes in them, more frequently. So if you're looking at R banding, which stains GC-rich regions, those dark bands would correspond to places where there are more likely genes. And those would be more likely euchromatic. There's been a lot of interesting things happening in the news recently. Not a political science class, but since we're talking about mutations, I thought it would be a useful introduction or time to spend a little bit of time talking about our genetics and how similar we all actually are to each other. And perceived differences don't necessarily have a genetic basis. Over the summer, one of the news channels asked me to help them figure out genetic ancestry of one of their news anchors. And so she approached me, she said, I'm taking these genetic ancestry tests, they're like $100 a pop or so, and I just want to know what I can find out about my ancestry, and I went ahead and took those genetic ancestry tests along with her, because I've got genealogists in my family, and I'm pretty sure, or I thought I was pretty sure where I knew where I was from, And it turns out that, yeah, not shockingly, my ancestry is mostly Western European and Scandinavian and British Isles. Big shock. And maybe slightly Iberian Peninsula, Spain, Portugal, and Gibraltar. Maybe not so much Gibraltar. So the question is, down here at the bottom, can we define genetically human populations? Is there any genetic basis to defining Middle Easterners and Asians, Hispanic? What do you think? Please.
I read once that people based in Africa are more prone to sickle cell anemia because it's supposed to protect them there from something. So that would be a way of testing the genes and the population and comparing those to other places.
So indeed, so sickle cell anemia, you've heard about probably forever in school, every other year or so in a science class. But indeed, there are mutations, differences, if you will, if you don't like the connotation to the term mutation. There are differences between us that help certain populations survive the climates and the regions they exist in. So yes, there are genetic differences between different groups of humans. But is that important? It is if you have one of these medically relevant mutations. But is genetic ancestry testing a hoax, or does it actually work? Why does it actually work? So there are genetic differences between populations of humans that can be useful for figuring out where you're from in the world. So here's kind of in broad strokes, one way this could happen. So geneticists agree that modern humans evolved in Africa, so 10 to 30,000 years ago. All of our ancestors, genetics tell us, were present in Africa. And of course, at the time, the continents didn't look as spread out as they are right now, but there were groups of individuals that left Africa and then went different directions around the world. And here's how genetic testing works and why microsatellites are important, is that you could imagine, this I'm making up. This is on real data, but I'm just using simple genetics to make the point. So you could imagine that there was one microsatellite in those ancestors of modern humans. They were living in Africa tens of thousands of years ago. They had 3 repeats of a TA at one spot on one chromosome. And then what happened as humans migrated around the world was, for example, in that group that was migrating through the Middle East and out into Asia and eventually colonized the Far East and then down into Micronesia and Australia, they might have had a mutation in their small group of humans that were in that group moving that direction that changed that to, I don't know, pick a number. Okay, so there's a mutation now, and that group of humans has TA5. So nowadays, did I just say nowadays? Currently, if you looked at that spot on the chromosome in people that were descendants of that group that was migrating through the Middle East and India, you might find that they all share that at that spot on that chromosome, we're talking about one locus, they might all share that mutation because they all descended from that group that had that mutation years ago. Now, what about the other direction, moving up into Europe and Scandinavia? Okay, so they might have had a mutation, a separate mutation because a different group of people, random mutation, they get a TA4. And then that spreads into the US during British colonization and so forth. So none of these mutations necessarily affect us medically. These are microsatellite changes, simple changes in length. They might not be in genes, they might be in the DNA that's in between our genes, but you can identify these differences that typify human populations. So the way that geneticists that work for these genetic testing companies do this is, and the way geneticists have for literally decades, is you collect people of ancestry that's a known ancestry. So they go to Africa and they go to the Sai or Mabutu people And they know that those people have been in Africa and their descendants for 30 generations. And so they collect a DNA sample from those individuals and they say, this is, we're going to define whatever your genotypes are as being typical of that population. And you go to the Han Chinese population, you do the same thing. You collect some reference individuals and you say, whatever's common there, we're going to say that's Han Chinese genotype. And Native Americans, and Siberians, and Inuits, and all sorts of populations. And so these tables that indicate the diagnostic genetic differences between human populations have been published. You can get them freely on the internet. And this is the basis for what, for example, the FBI uses. If any of you watch, I don't know, is it still on, CSI, and all those other procedural crime dramas, when they talk about, ooh, we've got a DNA sample, we're going to run it through the CODIS index, or the CODIS database. That's what they're talking about. This is a database of microsatellites. Those stutterings, repeats in simple sequence that have been defined for a number of genes or number of loci, the ones in yellow here, in humans. And here, instead of microsatellites, they call them STRs, short tandem repeats there at the bottom. It's basically the same thing as a microsatellite. So that's what the 13 CODIS core STR. When they have a DNA sample and they want to know who's it from, they're going to look at the genotypes of these 13 loci on different chromosomes in our genome, and they're going to ask, what are the genotypes? Then, they can look those genotypes up in these tables that I'm talking about. So I just want to introduce you briefly to this. Then we do a little activity. So here are some of those STR loci up across the top. So we've got CSF1P, they got funny names, FGA, TH01, and so forth. So the loci are the columns, regions of the chromosomes. And then the numbers in the table are the frequency, how likely it is that one person has any one of those allele sizes. So you can think about these allele numbers as, although this is not entirely true, you can think of those as number of repeats of a microsatellite. So if you have allele 5, that's TA, 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 TA. If you have allele 6, it's 6 copies of TA, and so on. So as I was trying to suggest, scientists that publish these sorts of tables go into, which population is this, by the way, those of you that are reading through this? Yeah, it says right here. So they say, they get DNA from a bunch of US Caucasians, and then they genotype a bunch of us, them, and they figure out how likely is it that each of these people have one of these genotypes. So let's pick an easy one. How about TH01 in the third column? If you were a forensic scientist and you had a DNA sample and somebody said it's from a US Caucasian, which allele would you guess would be the most likely one contained in that DNA sample? This column. Yeah, so you look for the biggest number. So what this means, 0.368, allele 9.3, means that 36.8% of US Caucasians have not really 9.3 repeats of a DNA sequence, but yes, 9 repeats, say. So 36% is the most common genotype for US Caucasians at this locus. So if you're a US Caucasian in the room, a third of us will have 9 repeats. Okay, what's the least likely one? Either 5 or 11. Right, so those are oops, wrong column. Those are really rare in the US population. But if you go look at the tables for US Hispanics or US African Americans or different groups, these frequencies change. And this is how forensic scientists can use our microsatellites to try to guess, when they have a DNA sample, for example, what ethnicity did that sample come from? Is that most likely left by Caucasian, Indian, Japanese, and so on?
So that table is specifically for US Caucasians?
Correct.
If you go to British?
Yep, different frequencies. This is a good question. So how refined do you have to be? in these tables to be accurate? Or are these tables accurate at all?
So, just out of curiosity, if someone was to say they ran someone's DNA through and it came out, DNA says that it's a male Caucasian.
Yep.
Can you determine age based on the gene?
Not age, no. So that's fortunate. Our DNA doesn't change over time. Well, for the most part. So that's something we can't tell. You can guess. You can predict or try to predict ethnicity with DNA data. And the take-home message I want to leave is not that, yes, ethnicity has a genetic basis, but that's not the differences that we see necessarily. That's just because of the way human populations have migrated around the world, and the fact that when we descend from somebody who had a particular type of mutation, we carry that mutation or difference with us. So it's more reflective of ancestral, historical migration patterns of humans. When you look at the diversity that's present in this room, as I think I mentioned on the first day of class, each of us is about 1% to 0.1% different. So we're 99.9% similar in DNA sequence, all of us. We're really similar to each other, and that's a beautiful thing, I think. There are some differences, they're not necessarily meaningful ones, but they're useful for scientific purposes. Okay. I'm going to distribute, this is in, let me describe this and I'll take your question. This is going to be sent out to you in a Google Classroom exercise. This is something that I'd like you to work on and be ready to discuss next class. Hopefully it's fun. It's a little bit of genotyping. It's going to use your knowledge about the definitions of microsatellites and using microsatellites to play forensic scientists to figure out who committed the murder. So while you read over this while you can, and then take the question.
Yeah, I was just gonna say, is that data reliable then, since only 1/3?
So this is exactly where we're heading with this exercise. How reliable are those microsatellite genotype data? How sure is the expert witness when they go into court and say, yes, I analyzed the DNA sample and I'm 99.9% sure that this sample matches the person that's on the witness stand? What does it mean that they're 99.9% sure? Is that enough to convict?
Human error could always play into,
Well, is there human error in genotyping?
not in genotyping, but in the handling of the genetic material. That's where the 0.01 gives them a way out.
We're going to talk about what that 1% difference means. When you say you're 99% sure, we're going to talk about that. And it has entirely to do with that table of genotypes and the frequencies of the genotypes. Yeah?
But I thought when they do paternity tests on someone, they won't say you're 100% sure that this is your father or this is your mother. They'll say that you're 99.9% sure.
So why is paternity testing not entirely accurate? Or why can they never say I'm 100% sure this is or is not your child?
Because of the Y chromosome in comparison to the X.
Haven't heard quite the right answer yet. There are many answers. One of them is new mutations can happen. So you could have a kid that doesn't look, for example, that is not entirely genetically identical to either parent because that kid has a new mutation that makes them look totally different. And would you say, sorry, you were adopted because your DNA doesn't quite match the parents, or do you conclude that, oh, it was a new mutation? And that's why there's always uncertainty, is that new mutations can happen, and there are always reasons why a sample doesn't completely fit our expectations of a match. We'll talk more about that as we go through class as well.
So when they say you are the father,
Like Darth Vader?
you still have that 0.1% that possibly you're not the father?
Right. Unless you know, like you were there. Which usually happens, but maybe not. There have been some interesting, genetics in society, there have been interesting news reports over the last couple of years about, I'll just throw it out there. Sperm donors getting used repeatedly when they weren't supposed to be used repeatedly, and who donated the sperm, and some interesting genetic testing that showed that there were some strange patterns of fertilization happening at like sperm donor, like one sperm donor was, he was like, oh, I don't know, I shouldn't make it up. He was running the sperm bank, and he wasn't actually using the sperm that he had told people he was using to fertilize their oocytes, he was using his own, or something like that, right? So Google it, see if you can find something interesting to talk about in genetics and science.
My question was for your comment earlier.
Okay, hold on.
I thought you had said that are
Telomeres.
Yes.
Right, so can you actually determine age by genetic testing? Telomeres, there are some companies, and I have some great slides about that, but I don't want to bore you with great slides all the time. There are genetic testing companies that claim that they will measure the lengths of your telomeres and tell you how much longer you've got to live. And those sorts of things. Jury's, I think, still out on the validity of those sorts of approaches.
But could they tell how long you already live based on the length that they should be and when they?
They would need a basis. though.
Yeah, they don't. So
Starting
they
basis.
can't. Yeah, they can't tell how old you are.
They would like your cell phone.
Yeah, exactly.
From when you
So
were a
that's why the
baby.
jury's still out. It's like, well, if you can't tell how old you are, how would you know how much longer?
Isn't
I'm-
it telomere, oh, sorry, you were.
I was just gonna, I was probably some of what you were, I was just gonna say like at some level, just because the length, calculating how fast it would degrade with placement and everything,
Well-
all that stuff, how long.
The length of your telomeres varies for a number of reasons,
It's not long.
including environmental,
But if
potentially.
you're like super
So,
stressed, I know that stress like makes you age faster, right? In like physical appearances.
but not necessarily telomere length. Telomere length has entirely to do with how many cell divisions. Every cell division, your telomeres get a little bit shorter. So your chromosomes shrink and shrink and shrink and shrink. And eventually that shrinking starts eating into genes that are near the ends of your chromosomes. And that's potentially what causes some types of aging.
So would the air apocalypse of Beijing affect young adults' DNA due to something like that?
We'll definitely talk about that when we get to environmental impacts on our genes. So I'm sending out this DNA fingerprinting assignment right now. And I hope you'll take a look at it for next class. I'm going to start a new Socrative quiz before, so two more things. One Socrative, this is not a quiz, this is just for my feedback, so I'm doing this one anonymously. I'm not asking you for your name or for your student ID number. I just want feedback on how you think class is going so far, if you're willing to give it. You don't have to. I'd love to hear your responses to the 3 or 4 multiple choice questions I have. And then for next class, we're going to have a few videos that I'd like you to watch. And we've got this Google exercise I just sent out. That's not going to be due to be turned in, but we might discuss it in class. Okay, so please work on it, but we're not going to turn it in. And then please, as I requested after class last time, send in some practice questions for the study guide. So let me launch that Socrative activity. And then I will see you next class.